If we had had more time during our photosynthesis lab, we would have conducted a pigment chromatography experiment in order to examine the different pigments that are present in the plant leaf. So in order for photosynthesis to transform light energy into chemical energy, the plant uses chlorophyll pigments to absorb and transform that light energy. We also see that there are additional pigments that are present in the leaves of plants, like carotenes and xanthophylls. And these pigments function to absorb light energy and then transfer that light energy to the chlorophylls. So chromatography is a process where molecules are separated based on their different solubilities in a solution that's called a solvent. So with paper chromatography, we see that a strip of paper is marked with a substance. In this case, the pigment extracts from a leaf, which we see here. Those are indicated by the leaf stain in this diagram here. Then that strip of paper is suspended within a tube that contains a chemical solvent that's located at the bottom of the tube. We see that the solvent absorbs into the paper and travels up the paper, carrying the different pigments different distances up the paper. And the distance that that pigment is carried depends on the solubility of that particular pigment. We see that carotenes will generally move the furthest up the paper, followed by xanthophylls, and then chlorophyll A and finally chlorophyll B will not move particularly far from the original placement of the leaf extract. So to calculate the RF value, we will divide the distance that the pigment moved in centimeters by the total distance that the solvent moved. The RF values will always be a decimal number between 0 and 1, with the larger decimal numbers indicating that the pigment moved further up the paper, while the smaller decimal numbers mean that the pigment did not move very far. So I've set out some example calculations of some RF values here using some example data. So here we see that there's a table that's laid out by the pigment type on the very left here. Second from the left is the distance that the pigment moved in centimeters. The third from the left is the distance that the solvent moved in centimeters, and then finally the RF value calculation. So in this example here, carotenes have moved 7.5 centimeters up the paper, while the solvent moved 8 centimeters, so we simply divide 7.5 by 8, which gives us 0.9375. We see that xanthophylls in this example moved 5.3 centimeters up the paper in comparison to the 8 centimeters that the solvent moved, so 5.3 divided by 8 would be 0.6625 in this example. Chlorophyll A moved 3.7 centimeters. Meanwhile, again, the solvent moved 8 centimeters, so we would take 3.7, divide that by 8, and we would get an RF value of 0.4625. And then finally, chlorophyll B, which did not move very far up the chromatography paper, moved 2.8 centimeters, while the solvent moved 8, so we take that 2.8 divided by 8, which gives us a RF value of 0.35. So using those example RF values, we can put those RF values in a table. So from the actual data that I will give you here in a moment, you will use the data and plot it in Excel in a table that looks somewhat like this, and then you can use that data table to then create a bar graph of the RF values. So here's the actual data for your RF calculation with all 4 pigments, again located on the very left of this table here, with carotenes moving 7.2 centimeters up the paper, xanthophylls moving 4 centimeters, chlorophyll A moving 2.4 centimeters, and finally chlorophyll B moving 1.1 centimeters. In our actual data, the solvent moved 7.8 centimeters in total, so in this example you would take, if you wanted to find the RF value for carotenes, you would take 7.2 and divide it by 7.8, and that would give you the RF value for that. For xanthophylls, you would take 4 centimeters divided by 7.8 centimeters, because that's the total distance that the solvent moved, and that would give you the RF value for xanthophylls. Same for chlorophyll A, take 2.4 centimeters that the chlorophyll A moved, divided by the total distance that the solvent moved, which is 7.8 centimeters. That'll give you the RF value for that. And then chlorophyll B, you'll take the 1.1 centimeters that the chlorophyll B pigment moved and divide it by the total distance that the solvent moved, which was 7.8. So you'll then take those data, put it into a chart in Excel, a data table in Excel, and then create a simple bar graph out of the pigments and the RF values. So the hypothesis to be tested in your lab report will be that all pigments will exhibit the same solubility and have the same RF values.